Okay, so now we're still learning about the central dogma of biology. And in this video, we're going to learn about transcription. So let's take a step back and understand what's going on right now. So we have a book. Let's just say, here's a book. And this book is called Bob's Information. And this book contains all the information that until, you know, it says Bob is going to have brown color eyes. Bob is going to have black color hair. Bob is going to have white skin. Bob is going to be a certain height, let's just say. Bob is going to have these genes or not these genes, and so on and so on and so on. Basically, you know, once again, DNA is our genetic material. And DNA will eventually code for a lot of these things in our body, which are called as proteins. But this book right here is DNA. And this book is coded, meaning that you can't just pick up this book and start reading like you read a normal book. This book is written in another language. You only can speak and write and read English, but this book is written as Chinese, okay? Let's just say that, okay? So this is DNA. The importance of transcription is to now take this Chinese language and convert it into English so you understand. Remember, you're the manufacturer, okay? Manufacturer in this case is a ribosome. What's a ribosome? The ribosome is an organelle that synthesizes proteins, okay? So you're an American manufacturer, and obviously you don't know how to read Chinese instructions, so you get someone else to translate this information, well, transcribe this information into English. So this Chinese language will be transcribed into the English language. So finally, you can now manufacture, in this case, proteins, because you know exactly what's going on. And this process called transcription, where we take DNA into mRNA. mRNA stands for messenger RNA. And this entire process of transcription occurs in the nucleus of the cell. So here is my cell. Here's my nucleus. And remember, once again, replication occurs in the nucleus. Transcription also occurs in the nucleus. And here I have my DNA, let's just say. And now we can, now we can actually understand the process of what happens in transcription. So let's draw a diagram. Okay. So I like to say that transcription is usually easier than understanding replication. Because in replication we have a lot of things going on. Transcription is a little bit more straightforward. So let's just say, here's my DNA. And I want to go ahead and transcribe a specific segment of this DNA sequence. Let's just say this over here. I want to go ahead and transcribe this specifically over here because this code, this just says, this codes for eye color, let's just say. Well, what happens, okay? The first thing you need to understand is that near the upstream of 5 prime, near the upstream of 5 prime region, there's something called a promoter region, okay? So let's just say over here, there's a promoter region. And in the promoter, there's this very specific small sequence called the TATA sequence, T-A-T-A. It sounds a little bit weird. It's called the TATA box, which is in the promoter. And you might say, hey, well, why do we care about this TATA sequence or TATA box in the promoter? It's because a very important enzyme will actually go ahead and come in the TATA box region. So let's just for now note that I have a promoter near the upstream of 5 prime region, and in my promoter I have something called the TATA box, which just stands for TATA. And obviously I want to go ahead and transcribe this segment over here of the DNA. What needs to happen? Well, similarly, just like in replication, I need helicase. So helicase is going to unwind the DNA strand so a big enzyme can actually fit in and go ahead and do some things, okay? So we just care about this top strand over here. This strand is going to be noted as our template strand or the sense strand. So here's my template strand. or my sense strand, and here's my anti-sense strand. Here's my non-template strand. Remember, I'm going to be generating mRNA, which is only single. It's not double-stranded. It's single-stranded, and it's going to be coming off from this template strand over here. I'm going to be transcribing this strand over here, so we don't really care about this strand. So what happens? So let's just say Helicase comes in. Now, don't worry, there's not 1,000 different enzymes like in DNA replication over here. It's just Helicase and one more thing. So I have Helicase unwinding, and I'm not going to draw another diagram because it's going to make it more complex. Let's just see what happens after it gets unwinded. So we have an enzyme called RNA polymerase. Just like DNA polymerase 3, how it comes on and binds the primer and starts to synthesize from the 5' to 3' direction, this RNA polymerase is pretty much the same thing. The RNA polymerase can only recognize, or really needs to bind onto the promoter region, the top box. However, RNA polymerase is also not that smart. It's a little bit dumb. It needs the help of certain elements. And one of the elements that helps this RNA polymerase become recruited to the promoter region is transcription factors. Transcription factors, okay? And there are specific transcription factors that help RNA polymerase come to this region. TF2D, okay, that's just as an example. You don't have to know this. Okay, and there's other transcription factors that help recruit RNA polymerase to the promoter region. What you do have to realize is transcription factors are aiding this RNA polymerase to go ahead and bind to the promoter region so it can transcribe this message. Okay, so transcription factors are promoting transcription in this case. But also we have another very interesting element called enhancers. And maybe I'm just going to draw enhancers as triangles over here. This is an enhancer. And enhancers also promote transcription. It's aiding for this RNA polymerase to come in, and it's promoting transcription to occur. And you'll notice something very interesting is that transcription factors are over here near the promoter region, trying to recruit this RNA polymerase into the promoter region. However, enhancers do not need to be near the near upstream of 5' region. We can find an enhancer over here. We can find an enhancer over here. And no matter where they are, they can still be aiding in this whole process. So, enhancers do not have to be present near the 5' region or upstream of 5' near the promoter. It can be anywhere. That's the beauty of enhancers. Enhancers are actually located on introns, non-coding regions of DNA. But we'll talk about that later when we keep talking about transcription. So, what's happening so far? Very simple. Helicase unwinds this. RNA polymerase, not so smart. It needs to be recruited to the promoter region. How do we do this? Transcription factors and enhancers. And finally, this RNA polymerase comes in, and sits on it. And now, finally, what happens? We can now transcribe the messenger RNA strand over here. Okay? And it knows, okay, it finally did that, and I have a single strand of mRNA, and finally this mRNA can leave, and then of course this DNA strand will reanneal with this DNA strand, and now we just have, this is going back to normal. But now we have to focus on this over here. This strand over here is called the mRNA strand. It's not really called the mRNA strand. I like to call it the pre-mRNA strand. Your textbooks also call it the pre-mRNA strand because it needs to undergo something called RNA processing, okay? So this is not ready yet to be exported out of the nucleus and into the cytoplasm. This undergoes RNA processing. Why? What happens in RNA processing? Why is this pre-mRNA? Why is this just not messenger RNA? Well, let's see what's happening here, okay? Over here, where I don't have boxes, these are non-coding regions of DNA. These are non-coding regions of DNA. Write this down. Non-coding regions of DNA. These are called introns. Very important. These do not code for something. These are non-coding sequences. Non-coding sequences. The boxes on this hand are called exons. These are coded messages. What do you think we want on our messenger RNA? Do you think we want exons or introns? Do you want coded messages or do you want non-coding messages? Obviously, you want the coded messages. You want exons. Remember, when your ribosome is going to read this mRNA, this messenger RNA strand, it needs to be coded with information so the ribosome can say, okay, hey, I'm going to make this amino acid, I'm going to make this amino acid, I'm going to make this amino acid, and finally make a protein. So exons are coding regions or coded DNA, and these over here are non-coding. What needs to happen? It needs to undergo RNA processing. So something called spliceosomes can splice out the introns. So we have spliceosomes that splice out these introns, splice out these introns, splice out this intron, splice out this intron, splice out this intron. So we can have a very, very nice mRNA strand with just exons. Just exons. So what do you end up with over here? You end up with something like this. 1, 2, 3, 4, 5 boxes. Okay, and it looks something like this. And it's all coded. This is exons, okay? And these are all exons, and these are coded. So what's the difference between pre-mRNA and the mRNA strand over here? Is that the pre-mRNA strand contains introns and exons, and the mRNA strand over here contains only exons, only coded information. And this happens by something called spliceosomes. I want to take a minute just to digress from this topic a little bit because it's very important. If you're taking an AP Bio course, it's very important to understand the importance of RNA processing and what it promotes. RNA processing promotes variation. How? Well, you see the spliceosomes over here. The spliceosomes don't necessarily have to cut out this intron, and this intron, and then this intron, and then this intron, and then this intron. Spliceosomes can actually cut out introns in any order they please. And by doing so, by splicing out this first, and then this, and then this, we're getting a different kind of mRNA. We're getting a different kind of final mRNA. So it's very important to understand that spliceosome, when they splice out introns, this is another mechanism of variation in our body because we're able to produce different transcripts of mRNA. Very, very fascinating. So now let's go back. We have this mRNA from pre-mRNA. We've undergone RNA processing, and we only have to do two last things. So what we have to do is add 2 slight modifications to this mRNA strand. Something else you actually have to memorize. And the additions we add that we add here are something called, this is not negative, a 5' GTP cap and a poly A tail. So this poly A tail actually contains a polyadenylated sequence of just AU, A, and so on and so on and so on. This poly A tail is their 3' end, and there's a 5' GTP cap. Why do we care about this? Because the poly A tail and 5' GTP cap stabilizes mRNA and prevent degradation. Very important. This poly A tail and 5' GTP cap prevent degradation. Remember, it's going to be in your nucleus. You need to export this out into the cytoplasm and finally meet a ribosome. So you need to make sure that this doesn't get degraded for some reason from going from the nucleus and exported out into the cytoplasm. So you must need a poly A tail and 5' GTP cap. And it's also very important to make sure that this mRNA strand, the coded sequence, is protected. So this is basically transcription. I'm going to go over it just in a minute to give you a very brief overview if something is not clear yet. We have a DNA strand here. Well, we have a double-stranded DNA here. We want to go ahead and transcribe this specific portion. What happens? Helicase comes in, unwinds its DNA strand. RNA polymerase can finally come into the promoter region, but it's kind of stupid. It doesn't know where it's going. Well, with the help of transcription factors such as TF2D and such other elements, such as enhancers, finally RNA polymerase can come over here, transcribe this mRNA strand, and now we have this over here. We have exons and introns. what happens? These introns need to be spliced out by spliceosome. We undergo RNA processing. Finally, we have this nice mRNA-coded strand, and we have to add 2 slight modifications, which is the 5' GTP cap and a polyadenylation sequence, which is called a poly A tail. And now finally, this is ready to be exported out into the cytoplasm. Now, there's something else very important that you need to realize is, remember the base pair rule? Remember the base pair rule for DNA? We have A pairs with T, C pairs with G. These are hydrogen bonds. Remember, A pairs with 2 hydrogen bonds, C pairs with 3 hydrogen bonds to G. Let's take a look at transcription, because transcription is a little bit different in that there is no something as thymine, there is no something as T in mRNA. So, write this down. So we have a very slightly different base pair rule. For transcription, we have the base pair rule, A paired with U and C paired with G. U stands for uracil. If you want to write this down, write it down. A pairs with U. A does not pair with T. So, what does that mean? Let's just say, here's my template strand, and here I have C, G, G, T, A, and my RNA polymerase comes in and synthesizes or transcribes a messenger RNA. What am I going to have here? C, G, G pairs with C, G pairs with C, T pairs with A. Don't get confused. Some people are like, hey, well, you just said we can't have any T. Well, no, I said the mRNA can't have any T. The DNA can have T, sure. What happens when we have a thymine here? I still pair with adenine. So I pair with adenine. Over here, this A pairs with not T, but U, uracil. So U and U. The last thing I want to talk about before I end this video is something called a codon. So a codon, and this is one codon, this is another codon. A codon is a sequence of 3 bases. So GCC, this is one base, 1 nitrogen space, 2 nitrogen spaces, 3 nitrogen spaces that code for a single amino acid. Once again, a codon codes for a single amino acid, and a codon is comprised of 3 bases. So the next video when we do translation, you'll see that you have a chart where you can easily look up online, and you'll see that, oh, GCC codes for a specific amino acid. And this amino acid will obviously be useful when we actually enter into the ribosome, and the ribosome can link these amino acids together. So just for this video, just understand that this is a codon, and a codon codes for an amino acid, and a codon has 3 bases. That's all for this video.